Biologists have been closely studying Darwin's finches in the Galapagos Islands, observing them evolving in real time. For over 4 decades, biologists Peter and Rosemary Grant have been investigating changes in beak size, beak shape, and body size that have occurred in finches native to the Galapagos Islands. Because the island of Daphne Major of the Galapagos is so small, the Grants team has been able to catch and measure all of the medium ground finches in the island's population, marking them with colored leg bands. The medium ground finch makes its living by eating seeds. Finches crack seeds with their beaks. Beak size and shape and body size vary among finch individuals, and these traits have heritable variation. So parents with particularly deep beaks tend to have offspring with deep beaks. Large parents also tend to have large offspring. Soon after the team began to study the finch population, a dramatic selection event occurred. In the annual wet season of 1977, Daphne Major received just 24 millimeters of rain instead of the 130 millimeters that normally fall. During the drought, few plants were able to produce seeds, so 84% of the medium ground finch population disappeared. 2 observations support the hypothesis that most or all of these individuals died of starvation. The researchers found 38 dead birds, and all were emaciated. None of the missing individuals were spotted on nearby islands, and none reappeared once the drought had ended. The research team realized that this die-off event was a natural experiment. Instead of comparing groups created by direct manipulation under controlled conditions, natural experiments allow researchers to compare treatment groups created by unplanned change in conditions. The Grants team could test whether natural selection occurred by comparing the population before and after the drought. In this natural experiment, the hypothesis is that beak characteristics changed in response to a drought. The null hypothesis is that beak characteristics did not change in response to a drought. We can test the hypothesis by measuring all of the birds in the population before and after the drought. The results support the hypothesis and demonstrate that natural selection did indeed occur. So why were deeper beaks adaptive? At the peak of the drought, most seed sources were absent. The tough fruits of a plant called Tribulus cystoides served as the finch's primary food source. These fruits are so difficult to crack that finches ignore them in years when food supplies are normal. The Grants hypothesized that individuals with particularly large and deep beaks were more fit to crack these fruits well enough to survive. At this point, the Grants had shown that natural selection led to an increase in average beak depth in the population. When breeding resumed, though, in 1978, the offspring produced had beaks that were 0.5 millimeter deeper, on average, than those in the population before the drought. This result confirmed that evolution had occurred. In only one generation, natural selection led to a measurable change in the characteristics of the population. Alleles that led to the development of deep beaks had increased in frequency in the population. Large, deep beaks were an adaptation for cracking large fruits and seeds. Characteristics like body size, and beak shape are polygenic, meaning that many genes, each one exerting a relatively small effect, influence the trait. So it can be difficult for researchers to know exactly which alleles are changing in frequency when polygenic traits evolve. To explore which genes might be under selection, researchers in Clifford Tabin's lab began studying the beak development in an array of Galapagos finch species, looking for variations in patterns of expression of cell-cell signals previously identified in chicken beak development. They hoped that homologous genes might affect beak development in finches. They found significant expression of a cell-cell signal gene called BMP4. There is strong correlation between amounts of BMP4 expression when beaks are developing in young Galapagos finches and the width and depth of the adult beaks. In these figures, the dark areas represent tissue with high BMP4 expression. On the left, you can see the expression of BMP4 in the tissue is, on average, lower than in the cross-section on the right. The left sample was taken from a finch with a shallow beak, while the right was taken from a finch with a deeper beak. When the researchers experimentally increased BMP4 expression in young chickens, they found that beaks got wider and deeper than normal. Similar experiments suggest that variation in alleles for a molecular called calmodulin, which is involved in calcium signaling during development, affects beak length. So alleles associated with BMP4 and calmodulin expression may be under natural selection in medium ground finches, suggesting a direct connection between natural selection on phenotypes and evolutionary change in genotypes.